My research field was bioinformatics. My name is Soberjot Singh. My mentor was Dr. Stuart Brown. My co-mentor was Jisha Wai. My other co-mentor was Jisha Mann. I did my research at NYU Medical Center, Department of Cell Biology. Bioinformatics, a little background. You guys already seen a couple presentations on bioinformatics. You probably might recognize some of these. A huge amount of data is generated from biomedical experiments. Bioinformatics uses computer methods to sort, analyze, and interpret this raw data. Bioinformatics works closely with technology labs that generate complex data with scientists who design experiments to study disease and basic biological processes. Basics. There's some background information. Unix is the operating system commonly used in bioinformatics. It's a very power-oriented OS, computer software. It's known to not be user-friendly because unlike Windows and stuff, you can't use the mouse to click on things. You just have to use code that's cryptic. Codes tell you. Okay. Perl. A program. Perl is a computer programming language. Perl is basically like, think of the computer as a foreign student, and Perl is the translator. So what you do is you write stuff in Perl, and because the computer language or a binary language is way too complicated and time-consuming for a human to understand and write. So we use Perl program to speed up the process. There's two kinds of programming. One is compile, which is an example of compile programming is C. So you first write the program, then you run it through a translator, then you run the program. The translator converts it into binary before you send it to the program. So basically, programs that are written in C run faster. So if you are worried about a time, you need to write it in C. And if you want simplicity, you write it Perl. NCBI is the National Database of DNA and Protein Sequences. This is an open source site. You can go on it and you can download research papers that have been presented or that have been published, and then you can have their research. So if you want to do more research on the same topic, you can go here and you can get the raw data and conduct your own research. So you don't have to get new data every time you want to do the same experiment. UCSC is the University of California, Santa Cruz. It's a national, this is a tool to visualize and annotate genomes. So you can actually publish your DNA software, like genes that you analyze, you can publish to the site, and you can go on the internet, you can look at your own DNA, and you'll have like peaks, there's like next diagrams. Or if you want to look at just genes in general, you can type in a gene name and you can pull up a database and it'll show you what the gene looks like, what chromosome it's located on. It's very crucial. BLAST. BLAST is the most commonly used software in bioinformaticians. This software is just basic, but it does vary a wide range of applications, but one of the things it does is just it compares data, DNA maybe, to a database of other DNA. So you basically say you have a questionable source of DNA, you just don't know what it is, you can use BLAST to compare to what it is, and BLAST will tell you it's the closest match to this species of primate, for example. That's just one thing. Source Storage is an open source software website. You can go on, there's not just bioinformatics software, you can get games, you can get anything that's open source. Open source, by open source I mean, like it's freely available and it's open to, you can edit it to the way you like. So that's what I mean by open source. So it's just software that's already published, it works. It might be a little clunky, it might not work for every program. Galaxy is a genome analysis. One of the things you need to know about bioinformatics is many of the tools you're going to need to work with are already published. So one of these toolkits is called Galaxy. You can use this to analyze your raw data into like more feasible results that you can understand it better. It's just some of the tools you can use. Okay. The next thing I want to talk about is this is a basic Perl program. The first line, that's called a path. So basically you're telling the computer to look in this location for the Perl translator, and then as it reads on to the next line, it'll continuously translate the English or the Perl into binary as it's running through the code. So you just, these are basically variables. You have to say my, and you don't have to say my, but you can say my if you want to. These are variables, so I guess it's kind of math logic. You gotta think of it in a particular way. This is a program I wrote a couple weeks ago. So this basically is just searching that the input data is in the right format. It's actually about 50 lines long, but I couldn't put the entire thing into a slide, so it's just the top part. Okay, next generation sequencing. Sequencing is a huge part of bioinformatics. About 80% of the jobs in bioinformatics right now comes from sequencing. It offers an accurate, efficient, and solution to costly sequencing. Generally, before this, you would use other methods that that were mostly taking place in the lab, and would take a lot of time. You would pay lab techs to work on with these expensive chemicals, which is incredibly costly. So sequencing is just like this giant box, and it's very complex. You just put your data in, it does its work, and you have your results. The applications for sequencing, what you can do with this is you can sequence genomes to identify mutations. So you can use it for cancer research. ChIP-seq chromatin immunoprecipitation sequencing. That's what ChIP-seq stands for. You can use it to locate and identify protein DNA interactions. So basically, when you have I'll get into it. Sequence RNA to measure gene expression. Gene expression is like, you have the human organism has about 30,000 genes, and not all of those genes are expressed. So how do you measure it? The more the gene is copied, the more RNA you will have of that gene. So the more protein you will have of that gene. And that's how your body uses genes. It turns into protein, and the middleman is the RNA. So this is the sequence at NYU Medical Center. We're going to get 2 more of these in the next couple of, I think next year. But this is the one we have right now. Those little vials are where the chemicals are. Just the little door, the gray door, that's where you can show the slide, and there's a camera inside. It takes pictures, and depending on, I don't know how it does it, but it looks at these dots, which is the DNA, and it determines if it's a base, which base, A, T, G, or C. There's 4 bases in here. I worked on various projects. These are not all of them, but these are the most important. In my last year, I did software testing. I did ChIP-seq software testing on Sizer and translocations with BWA, SAM tools, and breakpoints ready this year. Microbiome. This is a large project, but the part of it that we worked on was we worked with the NYU School of Dentistry with this project, and we used article classifier. That's another program that we did find off the line. It was already published, and it took a little while to get it running, but it works. Literature search and summary, tandem primers. This is actually a very interesting topic. It's still kind of in the background stage. It's not that important yet, but it's very, very interesting. There's not much research done on this. This is ChIP-seq. This is the entire process. You start with your inner DNA, and you have polymerase, POIs, and polymerase. That's where polymerase always bind to where the gene is. So you introduce enzymes and cut stuff into little pieces, and then you have antibodies that bond to the polymerase, and then you can separate it from the rest of the DNA. So you have just the polymerase binding site. So that's where the DNA, that's where the DNA and the protein interact. So that's a gene generally. So you put the DNA, you put the DNA into the sequencer, and you get an output. Like these are peaks, and so what you start off with is not one genome, not one cell. You start off with a whole wide variety, and the enzymes cut them up at various locations. They're not always at the same place. So when you put something in the machine, it locates the sequence that's cut up and matches it to a reference genome. So you can get peaks like, so the higher the peak is, the more likely the chances are of that location being an actual binding site for protein. So that's the genome. It's used, it's important. It's a genome. Okay. Software. Researchers and programs continuously focus on software. Open source software often does not work smoothly without the software. So a lot of times there's time has to be invested into making them work together, to find the right program to use and making two programs or any computer work correctly with another software. There are many programs developed to deal with the data text files produced by DNA sequencer. So the piece that you saw, that's not actually the final, the result from the sequencer. What you get is a DNA file, and that you insert into another program, which is it has a pretty simple job, but not a lot of them can do it. Not a lot of software can do this correctly. There's a need to validate and compare new software. So you need to know what works and what doesn't. This isn't unique bioinformatics. You can include the sites if you don't know what works and what doesn't. Software testing procedure. How would you go about testing a particular software? So you find out the one. Download a program on your computer to disable operating system. You find out what kind of operating system works on, and you download it. Get the program up and running. Most programs come with a test file. This is just to see if the program is actually working correctly, and they actually tell you what the results would be if the program is working correctly. So this is just like a test. Then you have to compare this program to another program that you already know works correctly. You want to see which one works better, which one's more efficient. So you get a file, because each file might have, there's many kinds of file formats, and not every software can work on multiple file formats. So you have to get a file that both software can work on, and you may use the format multiple times. Use a file which you already know the results of. So you might have a file that you already did the test on, you already use PCR and you know the exact results of that file, you know what it is. And then you run both programs onto that file and you get the results. And the results of the results that match the closest to the original is the correct